He's
Hello. First, I'd like to introduce myself. My name is David Costello, and just want to welcome everybody. And we're going to get the meeting started here shortly. I just would like to say that it is an honor to be part of this group. And the only announcement I really have is, we're starting a new management system for our securities, and there's an email sent out today. So if anybody has any questions regarding that, I'll be glad to answer them once the meeting is adjourned. I guess right now we'll just call the meeting to order, and Dr. Gulati will take over from here.
Thank you, Dave. And I would like to welcome. Hello, is that better? Okay, thank you. So, hello? I think it's on. No, now it. So it's coming in.
It works there.
Hello? Yeah. So, okay, so first of all, let me thank this kind. Maybe I should face this way, then it will. So, let me thank all of you for sparing your valuable time and coming here and supporting what we are trying to do. And there is a noble cause which we want to pursue forward. And before I can move forward, I'm going to introduce someone whom I have known since childhood till today. And he is in the audience, and he is not only my great friend, but an accomplished physician. He is the chief medical officer of many chain of hospitals. So I will request Dr. Marwa to come here and say something about our profession and what we are trying to do.
I think let me try to talk without the microphone. There's too much echo over there. So can you hear me in the back? Yes. Great. You're going to be hearing a lot today about the biology, the finances, everything that is so important in an organization, in a company. But I am going to talk about something totally different. I'm going to talk about why we are in this room and whose vision this entire thing is. As Anil said, on the way here I was thinking, I've known him for 54 years. It's not just the length of time that's important, it is also how I have known him. We went to the same school, St. Francis. We went to the same college, Christian college. We went to the same medical school, King George Medical School. We did an internship together. We moved to Chicago at the same time. So a very long friendship. And obviously, there's no one in this room, including Vandana, his wife, who has known him longer than I have known him. That being said, this is not an accident. This is not something because he accidentally walked into it. This is not something that happened just because he was lucky. This is a culmination of his vision that I have seen for a very, very, very long time. When we moved to Chicago, at that time there used to be a electronics store called Service Merchandise. That's when the Commodore computers first came out. He and I would go and buy the Commodore computers. We would literally stay up all night fiddling around with it, trying to make it do things it was not meant to do. And when we would mess it up, we would take it back to service merchandise, and without any questions asked, they would give us a new one. And I'm convinced to this day that we have part responsibility of making the store close down. When we used to be up late at night, he used to be talking. A lot of us talk. Most of us are just talk. How many of us have the ability to follow through on your vision? On the face of it, Anil was a very successful professor at UIC in the Department of Neurology. He became tenured at a young age. Most people at that time would have said, well, I've achieved my life's ambition, now let me just enjoy life. Not him. We would be up late at night talking about stuff that he wanted to talk about. In any organization, in any company, there are three things. What do they do? How do they do it? And why do they do it? Every company knows what they do. Most people know how they do it, but when you ask the question, why do you do it? They're dumbfounded most of the time. Anil entered into this organization with the why, not with what, not with how, with the why. And I'll give you my reasons why I say that. In the earlier part of his career, he was so passionate about artificial blood substitutes. When he and I were talking about artificial blood substitutes, he was not talking about that from a money perspective, a financial perspective. He was talking about the why. I need to work on artificial blood substitutes because if we have them, imagine how many lives could be saved. And in early part of his career, during that phase of his professional life, he actually partnered with the US Armed Forces because they saw the vision that he had in terms of the power of artificial blood substitutes. He worked very hard with that because of multiple reasons. That project did not end up where he wanted it to end up, and that was the biology of that product. After that, I saw his passion for a peptide that had just been discovered, endothelin. He was so passionate about it. And again, the reason for his passion for endothelin had nothing to do with the money part of it or making a company out of it. He would use me to bounce ideas off. Maybe endothelin is involved with hypertension. Maybe endothelin is involved with this or that and whatnot. He educated himself endothelin to the maximum, and then he became a world-renowned researcher in the field of endothelin. He's still active in that regards. I still remember to this day when the main can we remove that picture, please? It's not a good picture. I still remember that day when he first talked to me about Synthequin. He was using that chemical on rats. He came to my home, he was so excited. He was excited because of what he had observed happens with that chemical or with that agent in rats. And again, he was not talking about the what or the how, he was talking about the why. Look at this agent. What is the potential of this? In rats, it does this, it does this, it does this, it does that. That was his passion. Again, how many of us from passion come to this point where you have this august audience sitting in this room in Rosemont, Illinois? I don't even know what his official title is, whether he's a CEO or what, I don't know. But from my perspective, someone in his position has 4 qualities that are needed. The first one is vision. Obviously, the fact that we are here is a testimony to his vision. If it is not for his vision, none of us would be in this room. None of us would be where we are. Vision. Vision is good. But the second is the ability to bring together a team who can fix that vision. Just look around this room. Need I say more of his talents in that context, in that regards? Obviously, I don't need to emphasize the obvious. The third is the ability to inspire his team. Imagine when you see the finances, I'm surprised to this day how much has been achieved by this company by his ability not just to bring the team but to get the maximum out of them. It doesn't matter whether they're accounting, whether they're scientists, whether they're public relations, whether they're people who connect him with the moneyed people, I don't know. But in all of those contexts, he has been able to inspire those people to get to the next higher level. And most importantly is his ability to have learned from his previous experiences and not repeat the same mistake. I have seen that part of him. I was there when he was frustrated at some of his previous ventures that he was trying to do. He's learned from each one of those experiences. I think I'm done with my allocated time. So my purpose for here is that I am excited to be part of this organization, this company. I was one of the first people who believed in Anil, and I have been sitting on the sideline just admiring what has happened. So 3 cheers to Anil. Thank you so much.
See, I think let's forget about Mike. Can all of us hear? My friend, thank you so much. You are one of the great orators. I have to learn from you. And Chris is here. He is one of the communication person, and he always emphasizes. And Gary is here from California. So their emphasis is that you have a story, but to tell the story, there is a way to tell the story, and I have to learn. I have to keep on learning about that story, how to effectively convey that story. So once again, I thank each one of you for the faith you have put in. Without this, it would not have been possible. And I always tell a reason that, tell an example that I may have discovered, for example, in rat we got the findings. But the key thing is, as a physician, you have to bring those findings to humans. Then only you are serving the society. If you can't bring your findings to humans, then practically they are waste. I mean, you can generate a paper, you can generate whatever, but paper, there are millions of papers which are published. I mean, Jim McDonald is here, professor of ophthalmics at Loyola, and I'm an academic. We can find paper for everything, whether it is positive or negative in literature. The key thing is to have something and bring it to humans and serve the society. That is the key thing. So now if I am doing an experiment in rat to bring it to humans, I cannot do it alone. There's no way you can do it alone. The key thing is you all helped me in bringing to that level. And I'm very, very thankful for that. And I will start only after the gratefulness which you have and the confidence which you all have expressed in me to bring it to this stage. And I will quickly go through, there are mix of, because 70% of us are physicians, they will understand the science behind it, and there are 30% who are non-physicians. So I will try to be little bit quick, and if there are scientific questions which you can ask me later, and we can have interactive discussion as we go now. So let me quickly, we started the company in 2009. One of my postdoc who was here, Manish Nawale, I think you must have
heard that name. So I moved from University of Illinois into Midwestern University, and I had a compound which I worked 25 years ago in India. And then I told my postdoc that right now at Midwestern the infrastructure is a little bit compared to UIC, it is not that much. So let us use that compound, inject in animals, and we can see what is the response. So after 25 years of that compound lying in my drawer, we found that that compound was still very active. So that prompted us that, okay, there is something good in that. And then ultimately we learned that there is always blood lactate level goes down. Blood lactate is a very important indicator of mortality in shock. And previously from 1990 to 2000, I had worked on artificial blood, as Dr. Marma said, and ultimately at phase 3 it failed. So that was a setback. Army was involved, Baxter was involved, and I was the one who first time went to US FDA and got the permission for phase 1 trial. And so that time there were only 3 persons in Baxter division, and ultimately when it failed, there were 294 people, and practically everybody lost their job. I mean, so that was a mix. So that experience told me that this compound could be useful in hypovolemic shock. So I taught him how to create the model, and we created the model, and we found that it is effective. So we have to do dose response, that to find the right dose, and when we escalated or increased the dose, it became less effective. So then we started decreasing the dose, and 1/10 of the dose, which initially we thought, it became very, very, very effective agent for hypovolemic shock in rat. So that's how we started. Because I'm a university employee, so we declared it to university, and university has its committee for patents. So they evaluated the findings, and their committee recommended that okay, patent should be filed because of patentability, novelty, and commercial viability. So these are the 3 things which are important for filing the patent. So the committee decided, let's move forward. That is how this got started. Then after that, one after another patents, they started coming out. So we started the company. Then Ajit, I went to London. Ajit, he's a business person, and I'm an academician. So I thought that without collaborating with a business person, I will not be able to do anything. So I worked with him, and that is how we founded the company. Then he went to his people, we started raising the money, and that's how this formation of the company was there. So we have resuscitative agent. Then we later on discovered that we have a neurovascular disease 1620 for stroke, Alzheimer's disease, and right now we have 9 indications, a very strong and robust pipeline which is within the company. So, and you will appreciate that gradually as we are discovering more and more, there are a lot of strength which is coming within the company. So we have, out of 9, I will discuss some of these indications. First one is hypovolemic shock, second is post-operative pain, cardiac arrest. Cardiac arrest is a lot of interest, and I'll show you a little bit of data which we have. And diabetic ketoacidosis, diagnostic imaging of solid tumors, and then stroke, Alzheimer's disease, and spinal cord injury we have. And recently we started working on opioid tolerance. So I will go through some of these. Our business plan and financial strategy is that we are moving forward in India because the cost difference was so much that here to do a study, just for an example, we did a dog study for toxicology. So the dog study in India costed us, for Indian FDA, costed us about $25,000. And recently we completed for US FDA the dog study which costed us $277,000. So the difference is so huge, and affordability becomes a big issue. So we started as a small company. So keeping in that point of view, we thought, let us keep on moving forward. Let us do the science first, and if the science is good, everything else, humans are the same everywhere. If it works in one country, it will work in the other country also. So that was the philosophy when we started. So then stroke is a huge one. There is only one drug, RTPA to dissolve the clot, and only 5% of the patient can get it because of the time limit. 95% of the patients do not get anything, any pharmacological agent to treat stroke. Only thing is rehab and palliative. Whatever the symptoms comes, they get, that's the treatment. So here we have something very novel, and if this comes out to be successful, I think it will change the ground rules how we treat a lot of the CNS disorders. So we have completed phase 1, phase 2 is ongoing, and we expect by the end of just let me announce that for stroke we are doing 40 patients in phase 2, and we are blinded, and we cannot unblind till we reach the halfway mark. So till 2 days ago we were at 19, and today our 20th patient came. So now we are at halfway mark. So within 3 months of monitoring is needed. So we should be getting the stroke results in the next few months. So diabetic ketoacidosis is another issue which is high unmet need. Diabetic ketoacidosis develops, and then cerebral edema develops, and many patients die because of, there is 20% mortality if cerebral edema develops. So that's a very critical situation. So we are trying to work in that area. And we had a US FDA meeting in 2016, and we have the response. I'll share that response with you. Projections, I will quickly go through. Just in India, the projections are pretty, in the sense that we have about 2 million patients, price per dose, Ajit and we all worked on it, 250 number of doses per patient to 50% of the market and 55% margin, so 500 million, and that's at the peak. And stroke is even bigger, about just in India it can reach close to $900 million with a profitability of about 540. So the projected sales at the peak, I mean in US the company roughly about 2020 our target is to shock. We have achieved so much that we feel that we should be able to launch the product in December of 2019 or early to 2020. So we have a full year of 2020 to generate sales, and Ajit will get into details, but you can see the numbers and they grow very quickly, and shock is also approved in the subsequent months, maybe 4 to 6 months after that. So I feel from financial point of view the company will grow pretty big in 2020 and beyond. I mean 2023, just the Indian part is profitability of about 470 million. Another, now coming to science, numbers are, the things which we are working are first in class. Always I give an example of Lipitor is one which is first in class statins, and even today Lipitor is the one for cholesterol. That is how it is known. What we are working is all three are first in class products. They are not best in class, they are first in class. There is always a generic, then you have the best in class, then you have the ultimate thing is first in class, and that is what we have. So for example, many of you are physicians, and there is increase in cardiac output. So heart pumps more blood in a state of shock. So we want more blood to be delivered to the tissues so that the organs can survive. Otherwise they die and the death happens. So up till now we use vasopressors, we pump in lot of fluids so that the heart pumps more and more. 60 to 70% of our blood is pooled on the venous side and effectively is not circulating. 30-40% is pooled is on the arterial side, which is effectively delivering oxygen and nutrition to the tissues. What we have done for the first time, this is first in class, that we are increasing the cardiac output by increasing the venous return to the heart, and simultaneously most of the vasopressors, they constrict the arterial side. We are dilating the arterial side, completely different from what is present today. So very, very unique mechanism that we are increasing the output from the heart and dilating the arteries so that blood goes to the tissues very freely. So that's that. Then first in class on endothelin. Endothelin has become, my wife says that you are married to endothelin and I'm your second wife. So endothelin we have been working since 89, when 88 it was discovered, 89 I started working on it. And the whole concentration was, there is high amount of endothelin in the brain. But the whole pharmaceutical industry that time was working on endothelin. It was discovered in the blood vessels. So they were working to find out whether they can find an hypotensive agent to treat hypertension. And most of the trials failed, but ultimately led to the discovery of pulmonary hypertension. Today there are 4 or 5 products approved for pulmonary hypertension. But there's 100-fold higher concentration of endothelin in the brain. So from 89 we kept on working. We could not find any lead till about 2010-11, when we found that these endothelin receptors in the brain are very, very important for development of central nervous system. And once that discovery was made, we thought that let us use that technique to treat diseases. And that's how we started repairing the brain and formation of new blood vessels. So diabetes also, we use insulin, but many times diabetes, when patients, they go into ketoacidosis because insulin response is not there. So we found an agent which can enhance the response of insulin so that diabetes can be properly controlled. So all of these are first in class. So I'll go through manufacturing. Sridhar is here, Dr. Andorkar. He's the first one who synthesized endothelin, synthaxin for me. He went to India. That time the company was not even having test tubes. We went to the streets and bought the test tubes and made synthesis. And then ultimately the whole process of purification. Today we make 99.8% pure compound every time. Every time. So very stable, very, very good. And then we had to make it injectable. So Sridhar worked on it and made a salt out of it, and so that it can be dissolved within 10 seconds. We don't have, in critical care, we don't have time to, that we have minutes to dissolve. So we made that technology. It goes in solution in 10 seconds and we shoot it in the patient. So all that was done. So this is an example of a study which was done in Greece. So here 50% of the blood was lost. And to bring the blood pressure back to baseline, we needed 2.2 liter of fluid, and here only 300 ml. Mortality, 3 survived. All 10 out of 10 survived at 2 hours. 0 survived at 24. 9 out of 10 survived at 24 hours. And there was no neurological deficit in these animals. So these have been published now. This was the first experiment which we did, Manish did, in which we measured the blood pressure, and this is the tracing, and this is the mean blood pressure, and this is the heart rate. So blood was lost, blood pressure came down to 35 millimeter mercury, then we resuscitated. Blood pressure is going up. What is happening to heart rate? Going down. Tell me, any vasopressor can decrease the heart rate? No. So this was once again a unique finding, first in class, which told us that there is something different going on from existing vasopressors. Then we measured the left ventricle and we put the catheters, left ventricular pressure and volume both were recorded. And this is a tracing in which this width tells us about the stroke volume. So stroke volume is how much blood is pumped in each beat. So every beat, now when hemorrhage occurs, this will shrink. When the heart is not able to pump more blood in each beat, ultimately the blood pressure falls. So you will see the pressure keeps on falling down. And ultimately, these animals are coming closer to death. So you can see the heart has become very feeble. And now I will show you the next one. Here we gave, in this feeble heart, we gave the drug. And you will see what is happening. So gradually, very steadily the stroke volume starts increasing. So the venous return of blood is happening in the heart, and heart is able to, Frank Starling's law, heart is able to pump more and more blood. I mean, cardiologist. So you can see the pressure building up and the stroke volume. So we published these findings in American Journal of Emergency Medicine, where you can see that these are hemorrhage animals, and after hemorrhage, resuscitation with hypertonic saline, here all of them died, here all of them survived with the drug. So there is, and this is what this is another independent study done in Greece, where 50% of the blood was lost, and here you can see this is the control and this is the drug. Cardiac output very significant, and this is the vascular resistance. So once again proving that cardiac output is increasing, that means the heart is pumping more blood per minute and the arteries are dilated. So more blood can easily reach the tissues and supply oxygen and nutrition. Survival, none of the animal died with the drug. And so this is another, a different study came out of Greece once again, and you can see the venous pressure going up, proving that there is venous return which is improving. And once again, arterial pressure is going up with the drug, and vascular resistance going down, highly significant, and cardiac output going down. So multiple studies have been done to show that there is. So another important issue which happens in resuscitation is lung edema. We pump in lot of fluids, lung edema occurs, and many times death occurs because of two major reasons. One is cardiac arrhythmias, and second is lung edema and death. Here we measured the lung wet-dry weight ratio, and when we give the drug, you can see highly significant difference. Orovate's index, highly significant. Mortality, highly significant. So once again telling us that we can prevent cardiac arrhythmias from happening when we are resuscitating, and we can prevent extravasation of fluid when we are resuscitating. So that paper also came out, I think last month. So we like to publish. So we are different from other pharmaceutical industry which keep their findings secret. We like to publish. We like to have feedback from scientists, and that keeps on improving us. It helps us in designing the next and next. So this is one study in which we did the brain blood perfusion, and I'm trying to see the cursor. Okay, here. So we produced hemorrhage, and you will see the color change there. This is the actual laser Doppler camera which is measuring the brain blood perfusion. So you can see that going down. Perfusion has gone down. And when we resuscitate on this side, it will, I think now we gave the drug, and you can see the brain perfusion. So once again, we are producing a direct evidence that the perfusion, brain is one area where there is a lot of autoregulation. Kidney, blood flow, liver, and other you can improve easily. Brain is autoregulatory. To change brain blood flow is not that easy, but you can see that brain blood flow is improving after that. We did a lot of receptor studies and found that it acts on alpha receptors, and I won't bore you. These are basically, we know that how it acts. So we know that there are alpha 2B in the veins, they constrict, they return the blood, and we know in the brain there are alpha 2A which dilate the arteries, and that's the main mechanism of action. So details of mechanism of action we have worked out, and increased venous return and dilatation of the arteries. So what is comparable to the existing treatment? So at present there are vasopressors which are used. The big difference is they increase heart rate and we are decreasing heart rate. They increase peripheral vascular resistance, that means the arteries are narrow, here they are dilated. Increase in heart rate contributes to cardiac output, here it's the venous return contributes to cardiac output. There are isomers present here, there are no isomers, so manufacturing. So no evidence of arrhythmias and fluid extravasation because it does not act on beta receptors. So huge advantage over existing ones. So all the goals of resuscitation are achieved. Only thing is that vasopressors can be stored at room temperature. Here it is, the normal fridge is required 4 degrees temperature. Then they are stable for, we have found that 2 years stability has come out very good. And still it is continuing. For 2 years it is stable. So shelf life can be 2 years easily, we can claim that, and later on it may be more. So publications we keep on doing. Now coming to 2016, we interacted with US FDA and told them the story. And they said that manufacture the compound not in India, in US, and do the stability studies. I can get that manufacturing done in matter of 2 weeks because I have the team in India who has made it. Whether he's right sitting here who made it for the first time. All I need is collaborate with a company and pay them $250,000, and my guys will come and make it for US FDA approved facility. It can be easily done. There is no technical barrier. Impurity profiling and all that, we have done it, so that's why I have deleted. That part is done. We have the data. Then they wanted more impurities, residual solvents. In India, we have done it. So that also is done. Stability studies, once you manufacture those batches, we have to study. So that we cannot do till those batches are manufactured. Toxicology studies, we got NIH money, and NIH funded us for US FDA, and we have completed those studies. So which are cross, they are all done. Metabolism studies, they are completed. So toxicokinetics is in progress. So basically a lot of things. So now we are going back to US FDA probably by the end of this year to ask them for the response that, okay, we want to do the trials in US, what do you need? So after all this we are completed, so we are moving forward. So low volume resuscitation, increased cardiac, I think I have discussed all these. Patent position very, very strong. Australia, Japan, India, China, European Union, Canada, US patent also will be issued. I have already received the allowance, and September, I think 9th is the September 10th or something. The patent will be issued and we'll do the press release. So phase 1 we have completed, and I'll show you phase 2 also is now complete. Phase 1 data. This is how we did ascending dose. We went from very small dose to the high dose, and we did not, this is the dose which we want to use. We did not see any toxicity here, no toxicity here, no toxicity 5-fold, we saw no toxicity 10-fold, 15-fold we saw the first sign of toxicity. Big safety margin. And many of you are physicians, tell me when you are norepinephrine, can you go even 3-fold higher, 4-fold higher? There is always toxicity. So norepinephrine, so it is very different from vasopressor. Then we did multiple ascending doses also. And the toxicity was hypotension because the CNS part dilatation of arteries took place, and that lasted for 1 hour and was resolved without any problem. Then there was small dryness of mouth and drowsiness. Now these toxicity in a dying patient are very, very minor. So, and that also 15-fold higher. So we are not even, safety is very big. Half-life 1.1 hour. We designed the clinical trial in such a way that the treatment which is going on in every hospital remains the same. No change in treatment. Only thing what we do is we add our drug on top of that treatment. So that is how there are so many centers, so everybody has their own style of treating. So they maintain their treatment. So there is, the complexity involves is when we do the analysis, for example, this hospital is doing a certain way, this hospital is doing another way. Now when the data is coming, the variability is likely to be so much that we might lose the significance of our compound easily. But I'll show you the data what came out. So these are the hospitals where we are doing our team. We have in-house every aspect of clinical trial which is to be done. Very, very good. I'm so proud of this team that Manish is here, who is the first one to do these experiments. Then Dr. Gupta, he is the chairman of the R&D committee in India. This is the team, and David Costello was also there. So this was the initial small team when we had first or second meeting. So this is a video which we are, the animation which we are preparing to
which may lead to a decrease in blood perfusion to the tissues, cardiac arrhythmias, fluid extravasation. Overall, the survival rate is very low with the presently available treatment. Shock is currently managed by increasing blood pressure using cristoids, colloids, and vasoconstrictors. The infusion of vasopressors needs careful titration. Their use causes an increase in vascular resistance and heart rate, which results in the rise of blood pressure. However, the use of vasopressors may have side effects such as an increase in peripheral vascular resistance, which may lead to a decrease in blood perfusion to the tissues, cardiac arrhythmias, fluid extravasation. Overall, the survival rate is very low with the presently available treatment. PMZ 2010 is an effective resuscitative agent by acting through a unique mechanism of action by stimulating alpha-2b adrenergic receptors. More than half of our blood is pooled on the venous side and does not participate in supplying oxygen and nutrients to the tissue. When PMZ 2010 is administered intravenously, it stimulates venous alpha-2b adrenergic receptors to produce venous constriction and return the pooled blood to the heart. This results in more blood pumped out of the heart with each beat, producing an increase in blood pressure. Simultaneously, PMZ 2010 produces dilation of arterial blood vessels due to reduction in sympathetic drive. In addition, PMZ 2010 appears to block alpha-1 adrenergic receptors on the arterial side of the circulatory system, thereby decreasing arterial resistance. Combined effect of stimulating alpha-2A and alpha-2B receptors leads to an increase in cardiac output and arterial dilatation by PMZ 2010, thereby enhancing organ blood perfusion and enhanced supply of oxygen and nutrients to the tissues. PMZ 2010 has demonstrated significant improvement in survival following hemorrhagic shock. It offers several advantages over currently used vasopressors. Concerns of potent vasoconstriction limit the use. PMZ 2010 in contrast produces arterial dilation to enhance blood supply to the tissues. The most common adverse effects of presently used vasopressors include arrhythmias, fluid extravasation, and ischemia, which has not been observed with PMZ 2010. Current vasopressors need constant intravenous infusion and needs to be titrated carefully, whereas PMZ 2010 is administered in 10 to 15 minutes infusion with no need for titration. PMZ 2010 has a high safety margin. PMZ 2010 does not act on beta-adrenergic receptors and has no isomers. This provides a significant advantage over current vasopressors because of minimal risk to produce cardiac complications. PMZ 2010 is effective in small volume and is not likely to produce fluid extravasation as reported with current vasopressors. It significantly reduces the amount of vasopressors used for resuscitation, thereby limiting their adverse effects. In summary, when PMZ 2010 is injected intravenously, it acts on alpha-2b receptors in the vein to produce venous constriction and acts on alpha-2a receptors in the brain to produce sympathetic action, leading to dilation of the arteries, thereby improving cardiac output and tissue perfusion. This is a novel first-in-class mechanism to resuscitate patients with hypovolemic.
So, I mean, we are still working on this video to improve it. So basically, this will be the message going out. So let me quickly go to, I want to wrap it up in about 10-15 minutes. Post-operative pain, it's the same compound. We have completed phase 1. So we can use, but the higher doses are needed for post-operative pain. The advantage is that it is non-sedative, so compared to dexmedetomidine and clonidine, which are presently used, they are sedative, and this is non-sedative, so the advantage is it can be used in ambulatory surgeries. So that's the big advantage. So, but because the dose is higher and it is closer to 0.1 milligram in humans, so we are not pursuing this. We are keeping this on the shelf because of the safety margin is less. Cardiac arrest, very interesting data came out. This is a model of in-hospital cardiac arrest. Out of hospital cardiac arrest, there is 90% mortality. In-hospital cardiac arrest is 40%, 35-40% mortality. Here in this model, 4 out of 10 died. And when we gave the compound along with adrenaline, all 10 out of 10 animals survived without any neurological deficit. This article is now published in Australian journal Heartland. And so we are, and the dose is same as hypovolemic shock. So we are very keen on pursuing this area forward for cardiac arrest. It will be a big step to move forward. Diabetic ketoacidosis, we found that the response of insulin, which comes here, from here, insulin is here. All we did was added our drug and it is back to baseline within the same time frame. So making insulin more responsive and ketone bodies, they are coming down. And their cardiovascular stabilization. So Japan, US patent, European, Australia, China, Canada patents are all issued. We had a meeting with US FDA, and once again, it is the same recommendation that you manufacture in US and do the studies in US and move forward. One good thing was we proposed that we will do it in children, because children high incidence of DKA, diabetic ketoacidosis. They said that if you are using in children, then you will have to do phase 1 in children. So FDA themselves suggested that do it in adults, which is a very, very positive thing. Rarely FDA gives you suggestion, they just give comments. So this was a very good suggestion they gave. So solid imaging tumor, we have the patent, Europe and US and India patent issued. We have given rights to company in India, they are doing phase 3 clinical trials. Stroke, that is where I want to, this gentleman in the middle, he is Professor Yanagisawa, who discovered endothelin when he was a Ph.D. student. We are very close buddies, we work with each other, and it was his findings, so we attribute a lot to this person. He was given professorship, he is even today the national professor in the academy, US Academy. And Japan government ultimately gave him such an offer that he is back in Japan now, but US government has still retained his professorship as the national professor of the United States. So we interact with each other quite a lot, and this is another team which we have. So he is himself looking forward to where we are going to take his discovery of endothelin in future. He looks forward to that every time. And this is one thing which we discovered, that day 1 of birth, the green stain is endothelin B receptor, which I told you that they are very high concentration in the brain. Day 7 of birth, 14 and 28 in a rat, they start decreasing. And that gave us some information that they may be needed for the brain to be developed. So when we got that idea, we started embarking on more and more studies and ultimately found that yes, that is true. Endothelin B intensity goes down with age, but surprisingly we found that in blood vessel it is going up. So once it goes up, that means we thought that the brain neurons are now maturing by day 14, which is equivalent to 1.5 year to 2 year of human child. So once they are matured, these receptors are no longer needed, so they start going down. And that is exactly, but blood vessels are needed because you need more blood supply. So that is where they are going up. So ultimately we found that ETB intensity goes up in blood vessels, down in the brain, and we correlated with nerve growth factor, these are factors which are important for the well-known factors for development of brains, correlates very well with nerve growth factor and the blood vessel, which is vascular endothelial growth factor. Once again proving that these are related to development of central nervous system. So we targeted Alzheimer's disease first. And this is a water tank. It's about 5 feet in diameter. Water is filled, and the green quadrant we put a stool. And that stool is 1 inch below the surface of the water. Then we put milk in water. Now water is opaque. So the rat, when we put, they are not able to see the stool. So first day they are wandering around here and there. Second day they learn. Fourth day, as soon as you release, they straight go to stool. So basically they have learned that where the stool is. When we produce Alzheimer's disease, they are not able to find the stool. When we treat with our drug. So once again telling us that there is recovery, there is improvement in learning. Now what we do is we remove the stool. So then the computer systems, they calculate that how much time they are spending in this quadrant. So that will tell us the retention of memory, that how much memory has been retained. And you can see this is normal in this quadrant too. When Alzheimer's disease occurs, it goes down, with the drug it is coming back. So they are not only learning, but retention of memory also is there. So then we worked on all the biochemical factors. I don't want to bore you with. So biochemically, we know that ETB goes up, vascular endothelial, the blood vessel formation goes up, the nerve growth factor goes up. So all those things are happening with Alzheimer's, and that's what the model which we have followed. So phase 2 is going on right now in Alzheimer's disease. So we will see the results in the next year probably. Then we also targeted stroke. This is the model of stroke. And when we gave our drug, you can see the big difference between the infarct volume. So stroke also, it is coming out positive. There is almost 85% reduction in infarct volume when we do 3-dimensional modeling. So once again, the receptors, the same receptors which are going up, which we found earlier. And this is an interesting, we incorporated a dye. This dye goes and incorporates into the nucleus of the cell. So when a cell will divide, we can see that now there are 2 nuclei and there are 4. So we can actually count the number of cells, and we found that when brain infarction occurs, brain has a natural tendency to multiply some of the cells, and that is hugely augmented by the drug which we are doing. So basically there are hidden stem cells in the brain, and these hidden stem cells are stimulated by this injection, simple intravenous injection, and huge multiplication of cells occurs. Then we produced diabetes and stroke. We found the same recovery, very, very good recovery. Survival is better, and even we actually injected PCAM to actually count the number of blood vessels, and they are also going up in both diabetic and non-diabetic. Functional recovery, all parameters of motor recovery were pretty good in these animals. Then laser Doppler flowmetry, this is the infarction, and 7 days later, this is the result between the 2 groups. This is control, this is the treated. So we can see that there is, and this paper, I think we just got acceptance. It should be published in the next few months. We did phase 1 clinical trial, and we found that this is the dose. We can go maximum dose, we can go this. And our projected dose is 0.3. So we got the approval. We started the phase 2 with 0.3 now. So phase 2, once again, the treatment which is going on, we do not disrupt the treatment. We continue with the treatment. We add this drug on top of. If there is neurosurgical intervention, those patients are excluded. So the team we have already described, patent position, US, China, Japan, European, Canada patents are all issued. And right now I'm preparing, along with Augusta Fernando, she is helping me to prepare the pre-IND meeting for US FDA. We are taking all this data for stroke, and we will interact with US FDA. That may be done in the next few months. And then hypovolemic shock will be the next one. Spinal cord injury. We found that there are hidden stem cells in the spinal cord. And how we found? This SOX2 is a chemical which only stains the stem cells. And Musashi is another substance which stains the stem cells. And you can see in the periphery and in the center there are hidden stem cells which are present in the spinal cord. We did a lot of experiments, found that there are stem cells in the center and periphery. So we created a model in which we produce spinal injury, and the muscle functions were lost from 22 score to almost 2. And then gradual recovery in the rat took place in the green one, and the red is the treatment, and this is normal. So you can see the visual effect of how much recovery is taking place in spinal cord injury is a very, very tough thing to treat. There is nothing which is. And the percentage, the black ones are the control and the colored ones are the drug treated. You can see the maximal weight bearing leg, left leg is on the colored one, and similarly on the right leg also there is significant recovery. We measured synapsin level to see synapses are formed, so yes, they are being formed. So compared to stem cells, although stem cells have already failed, the trials have failed, but the problem in stem cells is there are neurosurgical interventions here and immunosuppression. Here there are simple intravenous injection and stimulating the hidden stem cells in the spinal cord. ALS, we are trying for first in dog study. This is going on in collaboration with the Institute of Healthcare Innovation, College of Veterinary Medicine, and my college. So we are to dose 10 dogs, 3 have been dosed, and now 7 are to be dosed, then we have the results. So let us see what happens. I mean, this is once again a very, very critical situation in which so Jim and I have been interacting. There are many, many indications beyond this where we can use this compound to see the recovery of CNS disorders. So stroke, Alzheimer's, ALS, spinal cord injury, and these are. This today, I think, where is Amrish? He is here. He did this very simple experiment in which normally there is 20% oxygen in air, right? He produced primary cells and exposed them to 3.5% oxygen at 37 degrees for 24 hours. And this is what happens. They are almost gone. All he did was added this compound, and you can see in the picture the difference between. This is in Petri dish. What is happening in the in vitro system, telling us that there is something which is very important which is going on. So now we are working on the mechanistic aspect, that how all this is happening. So opioid we just licensed last year, and we are very aggressively moving forward in opioids. And the whole idea is that morphine, the same dose of morphine is producing this level of analgesia, and the same dose we can enhance by giving our peptide, almost double the response of analgesia, and then we can eliminate tolerance. So here, from here the animals have become tolerant to morphine. All we did was make a peptide complete, and Dr. Sridhar is also working on this project, and he has submitted an NIH grant. So the mechanistic aspects, these are all figures from Dr. Sridhar, who has worked on this mechanistic aspects there. So We have started the process of developing it as a drug. So toxicology studies are going on according to US FDA, although we are doing in India. Now we have resources because of help from all of you, financial resources to do according to US FDA, and that has already started, and I'm anticipating we should be doing human studies in opioid tolerance by December of this year or January of next year. Very aggressive. First in class, I always remind again and again, first in class. Now I will show you something very, very interesting. And this is the first time human results of hypovolemic shock. These are mind-boggling results. Even I was not expecting what came out. So we screened 113 patients in different hospitals. And out of 113 patients, 38 patients were included. So eligibility 130, 75 did not meet the eligibility criteria, so 38 were included and randomized. Control got 18, and PMC 2010 got 20, and out of which 2 were withdrawn by investigator, 1 consent was withdrawn, and here 1 was withdrawn by the investigator. So completed study 15 and 19. Demographics, you can see the age between the 2 groups are more or less similar. Number of doses to resuscitate 1.4, 1.11. So here more doses were needed in control. They are all getting standard treatment, all of them. Here the drug has been added, here it was not added. Hospital stay. Unfortunately, the treated group, our drug treated group, were more sick patients, and hospitalization was 14.6 compared to 9.2. But ICU stay from 14, only 6 days, and there ventilator. They were on ventilator for a lot more time compared to the drug treatment. Percent survival, 2 patients unfortunately died in the control group, no death in the treated. Blood pressure, this is the control and this is the treated. Monitoring till 48 hours. And this is the individual, every patient's response. And you can see that the response there, and you can see the response. When we did statistics, P is 0.0001. We have yet to see that level of significance in any of the trials in shock. Diastolic pressure, once again, very clear demarcation between control and treated. P value 0.0001. This is the highest level of significance you can achieve. Number of patients requiring vasopressors, once again 53 and 21, very significant increase in patients requiring vasopressors. Amount of vasopressors 22 milligrams compared to 1.2 milligrams. Blood lactate level, very interesting, 4.8, 4.09, 4.28, 1.5. And lactate is the most important indicator of mortality. And individual patient lactate. Here P value is 0.29, here P value is 0.0028. Now I want to draw attention to 2 patients. This patient 10 in control and patient 12 in the treated. And these 2 patients are of lot of interest because they are both females, and most of the time massive blood transfusion is needed either when there is a gastrointestinal puncture or excessive bleed, or there is OB-GYN, and because of pregnancy or so here molar pregnancy, here there was hysterectomy once again because of placental rupture. Both patients, one is 24, other is 31 years of age, young. And lactate level in one it is 11.8, other 17.41. 17.41 got the drug and survived. That is patient 12. And 11.18 is the patient in the control group and could not survive. Base deficit, once again, highly significant, individual values 0.001. Creatinine levels, highly significant decrease here. So once again 0.001. There also it is significant, but it is not more here. And that led us to another discovery that we can use this compound for acute kidney injury. So this is the glomerular filtration rate of individual values and acute kidney injury. Now Vivek is here. He is doing experiments. He is a critical care person. He is doing some experiments on kidney injury and renal blood flow. And when we give our drug, we can see the renal medullary blood flow going up in male rats, in female rats. And when we infuse norepinephrine, still the Doppler flow is up. This is the actual tracing which the experiment they did last week, I think. So this is a raw tracing, and this is, you can see the hemorrhage is produced, then we do the renal plan. So renal blood flow is almost gone. Then we resuscitate and with blood. So this is blood being infused, and then after that. So you can see the picture that is 10:33. So roughly about this is the time when this picture was there, and this is 12:30. So somewhere around here, this picture is. When we gave the drug, you can see the difference between this. So we have about 2 months or 3 months ago we filed the patent on this acute kidney injury. This is a new indication which we will be moving forward. So this is the summary of Amresh and Vivek again. So you can see the clear-cut difference between statistical value 0.01 in the laser Doppler perfusion in kidney injury. Now comes the interesting part. We have been naming it as Centhaquin. We call it as PMZ 2010, and all these names are gone, will be gone, because we have come to the stage that we are first in class, and Centhaquin is not a scientific name. Gaillard has been instrumental in selecting some of these. I mean, can you imagine that we are fortunate to give a name of the class like Lipitor was given, a statin name was given from somebody. And we are fortunate that we are here, so the suffix is Algron has to be given in this compound. So any future compound which is developed on this will have to name as Algron. And then this is the part we had to select. So if you like some of these, then give credit to Gaillard. If you don't like it, blame him. So, I mean, it is a learning process. Even I didn't know that you had to completely. So this is this has gone to WHO, and WHO will approve. And similar we have done for the stroke compound also. So as of this is about few months. I mean, I think 1 or 2 months back, 53. This is the capital structure of the company. Most of you know about it, and obviously we are doing first in class. We have achieved this. We have achieved preclinical validations. IND filings are done. Phase 1 has been completed. Phase 2, phase 3. We are we are already, that naming thing already tells you that we are preparing for product launch. And phase 2 data is so convincing that how can any regulatory agency, particularly in India, can deny the approval of this drug? I can challenge them that when you are saving lives, how can you deny the approval of this drug? So, and phase 2, phase 3 trials, we are doing in such a way that we will be going twice the number of patients in control, twice the number of patients in drug treated compared to control. Because ethically now there is a dilemma. When this is working, how can you deny not giving the drug if a patient is dying. I mean, you saw the examples. It's not one. I mean, it's one after another that we are seeing that. I mean, and there are OB/GYN people here. You can see 17 lactate level, and the patient survived. 17.4. So when the drug works, I mean, it is very, very big ethical dilemma that how you design a trial and not justify giving the drug which has shown in phase 2 that it is effective, that in phase 2 you are not giving it. So this will be a challenge. Let us see how it shapes up. And then the money part is obviously we have gone through this, and the company is going to become profitable. We invest in the company not only for money, but we invest to serve the society. And if in the process we can make money, it is good. And now I will request at this juncture, we have with us Mr. Ray.
He's right here. He's right here.
Oh, okay. So, University of I took this from LinkedIn, and he has been chairman of the board of Northwestern for many years, chairman of the board for 4 years, and board of directors for 32 years in Northwestern's memorial system and all the art. He has extensive experience in management, and I will appreciate if you can say a few words.
Dr. Anil again asked me to say a few words, and he knows that I'm a retired lawyer, so he has emphasized the word few. And I also caution you that I am not a scientist, and that all I've had is 1 year of biology, and that was in the summer at Northwestern because I was terrified of taking science at Georgetown with the pre-meds. I will say this, that I left the Northern Trust Company and started practicing law many, many years ago. And at that time I was on the board of directors of Northwestern Memorial Hospital. And I served there a long, long time. I served on various committees of Northwestern Memorial Healthcare, which is the parent. Northwestern Memorial Hospital is one of the subsidiaries. At that time we had about 9 or 10 subsidiaries. Recently we've had 6 before we started acquiring all these other hospitals. And I was chairman for many years of the hospital committee on quality, risk, and utilization management, which had oversight of the quality of practices of the physicians. And I served on a number of subsidiary boards of directors, including Northwestern Memorial Insurance Company, which was the Cayman Islands Insurance Company, Northwestern Memorial Foundation, which provided and made grants to researchers at Northwestern University Medical School for research. So I served in many, many capacities there. And on the legal side, I was doing a lot of work in healthcare because I kind of tied in with my volunteer work at Northwestern. And I did a lot of work in health information technology, and also did a lot of work with Pharmazz, including an early stage by the name of Norex, which was a Northwestern University affiliated startup. She was at an early stage. It ultimately was sold for about, I think, $100 million. I forget the name of the company that acquired it, but you can look it up as NAUREX. But I have to say, I only limited my area to the early stage of that work, and it grew and grew and grew, and put in some new management. So I've been around the horn a long time, got gray hair. I think what Dr. Neal is trying to do here is extremely impressive. He's got a great team, and I would be glad to assist in any way that I can at this stage of my life. So, congratulations.
Thank you so much. We have another special guest, Gary Carter. He has flown all the way from California, and we are fortunate to have you here. And
Thank you, Dr. Gulati. When Dr. Gulati asked me to come here last week, he didn't mention anything about speaking, so I was a little surprised when he hit me. And particularly after listening to some of the speakers today, not just the exciting news here, but is it, how do you pronounce it? You're a great speaker. I mean, I'd like to hire you to be my lead-in guy. But yeah, I've known Dr. Gulati not for 50 years, or I'm not a scientist, I'm not a lawyer, but I do have gray hair or no hair. So I hope I've learned something over the course of my career. And what is that? Well, in 1979, I was a MBA student at Harvard Business School. And the guy who lived next door to me came by one day and he said, "Hey, you're from Seattle. I have a friend that I went to undergrad with at Harvard who's starting a software company in Seattle. His name is Bill Gates. Maybe you want to talk to him." And I said, "Well, I don't want to go back to Seattle, but I called up a friend of mine, one of my oldest friends, and said, 'Hey, you should talk to this guy.' And he called me back a few weeks later and said, 'I can't find him.' This is 1979. Eventually he found him, and he ended up employee number 125. And then I stayed on after my MBA and worked with my doctor for 2 years in business strategy and management, development management. And I got to the point at the end of 2 years, as one does, where I've done my coursework and I've taught and I've passed my orals, and it's time to find a thesis topic. And I had programmed mainframe computers before I went to business school and built management systems. And I thought, well, there's this new technology called a microcomputer that could have a major impact on the way the world works, the way businesses are organized and managed. And I talked to the faculty about it, and it was not met with a lot of enthusiasm. So I took a leave, and my friend was now at Microsoft, so I went out and I met with Steve Ballmer, who had just joined as CEO, and I really didn't like Steve Ballmer very much, so I was not too enthusiastic about that. And there's another company that, most of you, everyone's heard of Microsoft. Most of you, except for a few who are early Commodore players, etc., haven't heard of a company called Digital Research. And this company had a product called CPM, and they were the first-in-class company in operating systems in 8-bit computers up until the time IBM came into the market. So that's where I went to work. And it seemed like a smart thing at the time because it's first in class, 80% market share in the microcomputer industry. And I spent 2 years there, and by the time I watched the company go from an 80% market share to a company that eventually sold to Novell for maybe $70 million, and nobody knows about it. And what I learned there, that wasn't an accident. I learned that it's not just about the technology, it's not just about the science. Management matters. So it was a really profound lesson because I saw how much it impacted the people who worked there. So I sort of went off on a mission to try to be someone who could be demonstrate great management in technology. And over the course of the years, I've just always been drawn to entrepreneurial situations. I love entrepreneurs. I love them for their intelligence. I love them for the way they can see things the rest of us don't see. They can take something from over here and something from over here and say, wait a second, what happens if I put these two things together? So I spent my whole career working with early to mid-stage companies. And I think if I had to put it in doctor terms, it's like the companies have a growth phase. They have birth, they have adolescence, they have the pains where they go to the marketplace all of a sudden, and what does that take? And then they have the pains that occur with maturation. And so I really work with companies through all those types of situations. It's a bit like being a doctor for companies. And it's something I enjoy a great deal, and my wife would sometimes prefer that I just went and found someone who just paid me a lot of money to work at a large company. But I love dealing with people like Dr. Gulati. I happened to be, I went to India for a wedding. One of my classmates at business school is a very successful person. And he has a couple of companies in Bangalore, and his son was getting married, so I invited some of us over for the wedding. And I was at the wedding, and I go to the bookstores. Bangalore was this, it reminded me of Silicon Valley, and when I first went there in 1982, this energy and entrepreneurship, and as I was, and I came back from that and said, I'd like to spend some time over there. It's both for the fact that it's, in these days and age, China or India, it's good to know something about one or the other or both. But the other is just the energy and the fact that it was an earlier phase of the development. And so eventually I found some people there, promoters, as we call them, who had a company that needed some help. And while there, I was having breakfast at a restaurant. I met this woman. We started talking, and we became fast friends. And she'd worked at IBM at one time. We shared this common fascination with, or attraction to, startups. And she called me up one day and said, there's someone you should meet. It's Dr. Gulati. And so I called him up, and we started talking. And I found myself coming back to him and talking because he's a wonderful person. I mean, I really enjoy talking about the science, but talking about his company, he's just about life. But that's not enough. At this stage of my career, I talk to a lot of entrepreneurs, and a lot of them want me to help them, thank God. But there's two things that I look for. One is the science or the technology, and the other is the people. And in the case of the technology, I have one classmate who's co-founder and was the first CEO of a company called Nectar. Nectar's current market cap, it's a biopharmaceutical company. Current market cap is about $10 billion, and he's currently the chairman. He teaches at Stanford Business School on entrepreneurship and healthcare management and so on. So I went to him and I sent him these materials and said, what do you think? And he said, first of all, are you crazy? Why do you want to do this? It's tough. Science, pharmaceutical company, biotech, it's tough, and most of them don't make it. And I don't really know too much about that space, but I went to another classmate was a VC, and he was a co-founder of Core Therapeutics, some you may have heard of. And he's currently a Ph.D. student at Berkeley, and I sent it to him, and he called me up and said, are you crazy? Why do you want to do this? But he's the kind of guy, Dr. Gulati sent me a PowerPoint of like 103 slides. And I come to a place that says no PowerPoint should ever be more than 20 slides, and hopefully 15. So 103. My friend Lee, looked at all 103 of them. And he, after he got finished telling me how crazy I was, he was saying, I don't see this, I don't see that. And he said, this is how I knew he looked at it. He said, look at slide number 93. This is where he organized his research facilities in India. Somebody knows what they're doing, because this is exactly the way I would have done it. And I've talked to various other people about it since then. And, so the science, I can trust in the science. But what I've gotten here tonight is I believe in what smart people decide to do. And I've met people here tonight who, it's very evident to me that they know what they're talking about. These are not casual investors. These are trained scientific minds who've looked at this. And I sent this to a friend of mine who's a emergency room physician. And he said, Gary, if this works, this can be amazing. I mean, this is really a problem. This could make a big difference. So I've got, but by being here tonight, if you people believe in Dr. Gulati, he's not just a nice guy. He's also a smart guy creating a potentially successful company. But I've also heard from people who've known him for like 50 years, and he's also a man of character. So it's not just about being smart. It's about what's your character, because I've seen that I've been screwed by or taken advantage of by lots of people in my life, because I like to be with people I trust, and to know it doesn't take you can get around almost any contract. if it takes a contract to get someone to do the right thing, I don't want to work with them. I want to work with them because if I turn my back, they will always do the right thing. And that's the kind of character that I've been hearing about tonight. So, this is I'm very happy to be here, to meet you, to be part of this. And I think that this is going to be a fantastic company. So at this stage, when I look at companies, it's how can I help? I don't have to be this, I don't have to do that, but any way that I can help, I'm here to help. So thank you very much.
And I'll quickly ask my co-founder to, let's not go in details, but this is an important plan because we are already embarking on a marketing plan. So, if you can.
Speaking after so many luminaries is a big tension, big job. Anyway, my job starts when the product is about to be launched in the market. And the vision is to make a full-fledged pharmaceutical company on a global basis. But since we would be launching this product first in India, and we are planning to launch sometime in Q4 of next year. So we will start the build-up process from June onwards, June 2019 onwards. And India is a big country in terms of marketing and distribution also. So we have divided India into 4 parts, and the operational team would be comprising of regional managers, area managers, and representatives. And since this product is of critical care, we would be building the demand through key opinion leaders. So our focus would be directly on critical care hospitals and the doctors who are directly responsible for prescribing these products. So this would be the total team. Marketing would be on a pan-India basis. And in order to build up the opinion with key opinion leaders, Dr. Gulati will do the technical presentations. And on the marketing side, we are experienced enough to handle the market in India, in the hospital sector, in the nursing homes, and in the critical care areas. Product would be sold mainly through institutions, not through private practitioners. Now, I think we have covered this in the business plan, but just for the benefit of everybody, the estimated market in India is about 2 million people annually on the hypovolemic shock. And in the first year, we are targeting 5% of the market and going up to 35% in the later stages. But as everybody and Dr. Gulati has mentioned, that this is the first in class, I would consider these projections to be on a very, very conservative basis. And this is the revenue potential. We have targeted a price of $500 with a net realizable value of $300 in the first year. 2 vials would be given to every patient. And the projected revenue on a year-on-year basis is, without increasing the price, is from 30 million going up to $227 million. This is for ischemic stroke, which we are planning to launch in 2020. Again, the market size which we are projecting is 1.3 million is the number of the patients. And first year we are projecting 2.5% going up to 17.5%. And this is the market size, and the price what we are estimating is 6 vials would be given to a patient. So we are estimating a net realizable value of $1,530 per patient. So the revenue generation is going from $49 million to $417 million from 2020 to 2023. So both the projections combined would give a revenue of approximately $700 million with a net, that was covered in your slide. So that is all. Thank you.
I'm glad to see that's the way it works in India, because when if you start calling in the United States, it's very difficult to find out who makes the decision and purchases. It could be the vice president in charge of supply chain. So it's a very different marketing environment here.
Yeah, in India also there are different sectors in which you can market the products. But since this is a hospital-only product and mainly for critical care, so we would be focusing on key opinion leaders.
My friend Gaylord, please.
I would like to not stand in the way of the gym. Number one. Number two, most of you who know me have heard enough from me. And I'm very grateful that you're here. And I'm very grateful that I'm here. And those of you who've not met me, I look forward to meeting you, and if you have questions that I can help with in any way, it'd be a privilege and a pleasure. I'd love to talk literally endlessly, but I don't want to take anyone there. But I will say, when I met Dr. Gulati, I'm at an age where I feel I'm entitled to have my own opinion and tell it like it is. And I had the opinion that this man is a genius. I didn't ask anybody about it. I made up my own mind. I worked with 40 years in real estate with, honest to God, I thought he was the hardest working man I've ever seen. And my old man was a hard working man. My partner for 40 years was a hard working man. I was in real estate finance in Chicago, commercial, for 40 years. When I met Dr. Gulati, I said, he must be running on adrenaline. Honest to God, I have not met a harder working man. I worked with Dave Costello for decades in real estate. We had our own firm. We were smart enough to get bought out by a bigger firm. Dave came with us, handled all of our company, all of our money, like it was his money. And we used to represent Cigna. I'll never forget when Cigna said, hey, a big institutional representing commercial finance, pension money, saying if you just treat our money like it's your money coming out of your own wallet, we'll get along fine. That was Cigna telling us how we would represent them as their fiduciary representative in Chicago. Well, I had confidence in Dave Costello. When I learned about this, I said, oh my God, you got to see this. I got forced retired out of real estate for a half a dozen years. The reality, I've been doing it for 40 years, enough. When it became impossible, I recognized that. I sat down. When I met Dr. Gulati, I first saw this 2 years ago. It was over my head. I saw it a year ago, and I really paid attention to that WebEx. Anybody who really pays attention to the WebEx says, there's a lot of science here. Dr. Gulati understands the science. And hey, if anybody can do it, Dr. Gulati can. I showed Dave Costello Pharmazz, said, please join us. When I met Dr. Gulati shortly after watching the first WebEx, he said, you're going to help me run this company. And I said, by God, the mission, saving lives, would make my life worth something. I can tell you, I believe in Dr. Gulati. I believe in what he's doing. I believe this company will do it. We all try and evaluate risks. We tell the story of Microsoft's founder. Well, the story of Zuckerberg, Facebook's founder, was sold his company to a venture firm at the very beginning. And when the venture firm saw this thing, opportunity, go like this, and he got $1 billion offer from Yahoo, they literally sat on him, made him take the deal. He signed it. Yahoo flubbed it. Failed to deliver the $1 billion. Facebook's $500 billion. why am I telling you this? A risk, in my opinion, this deal is taken an early buyout. He's been chased by people trying to say, here's $10 million, it's going to be a 30-year company. Other venture funds have made offers. I believe the company will be north of $1 billion. I believe it will have opportunities to take an early buyout. Dr. Gulati is a collaborative person. He's going to say, I want to see a show of hands. Very careful not to raise your hand at the wrong time. He's gonna, if you guys all raise your hand, he's going to be like Zuckerberg silent at the wrong moment because it's him. I asked him, tell me about each of the shareholders. He told me about every single one of the shareholders that are in it, that I didn't have anything to do with. He knows them, cares about them, and is committed to this mission. I feel the same way about my shareholders. I feel the same way about this company and its mission of saving lives. It's a privilege to be part of it, and if I can answer any questions at any time, I'm a phone call away. Love to hear from you, and thank you very much.
Gaylord, all I can say is I feel fortunate that many of us, we are closer to retirement, and Gaylord went into retirement, and then I brought him back. So I have the ability to bring people back from retirement. So be careful. Any questions we can answer, I'm there, Gaylord, David, Ajit, yeah.
Yes, you mentioned with 1620 the possibility of treating Alzheimer's disease. What about other types of dementia, specifically frontotemporal dementia?
Yeah, actually, the dosing schedule will change, and any type of neurodegenerative disorder, if it comes out to be successful, any type of neurodegenerative disorders can be targeted. But I'll tell you frankly, what is happening is the experiments which we are doing, when the damage is occurring, if we give the drug that time, the response is much better. So Jim and I the patent already covers, but Jim, he's an academician. Cerebral palsy is one area which I think this drug should work very, very good. So we do not have any experimental model which we have done, but down the line we will be doing those experimental models, and so the scope is very big, very, very big. So stroke, we are targeting that within 8 hours of stroke, this drug should be given. So it's not that it can be given anytime. Once the healing or scarring occurs, this drug will not be effective. So it has to be given early stages. So as soon as Alzheimer's is a progressive disease, so that's why I think it will work, because it's a progressive loss of neurons and progressive loss of ALS, also progressive loss. So in those kind of diseases, it is likely to work better.
We have to do 8 hours, RTP and all 4 hours, right?
Yeah.
So here we picked 8 hours.
We picked 8 hours mainly because we don't want 4 hours. We want to extend the time. So if it is 4 hours, then it becomes 95% once again will not be able to receive. So we did a lot of experiments in animal models, and we found that 8 hours in humans should work.
Yes.
What is the interaction of 1620 with stem cells? What's the mechanism there? It seems to be successful.
Yeah. So that's a very fundamental, very basic question, that there are hidden stem cells within our body, and particularly in the central nervous system. So the neural stem cells which we call, they are very unique. They divide into two, and one of them is a neuron and the other is once again the stem cell. And this one divides once again neuron and stem cells. So ultimately the number of stem cells are preserved all the time in our brain. They do not increase, but the neurons keep on increasing. So this drug is acting at fundamental level. Neuro D, there is a compound called Neuro D which is present in our body, which stimulates the division of these stem cells. And we found that Neuro D is the one which just goes up and the cells start dividing. So, and then there is a control mechanism which is notch signaling, which tries to control that extra neurons should not be formed. So in body there is always a balance between. So when there is damage, that's the time the proliferation occurs. So if there is no damage, this drug will not be effective. Yeah.
What's the mechanism of this drug working
On opioids, yeah.
on the It's a very hot topic.
Yeah, opioids are, the tolerance to opioids develop when there is G proteins are uncoupled. So you keep on giving morphine, but the signaling mechanism is lost. So more morphine, and more uncoupling keeps on taking place. This drug somehow just recouples in one shot. It just recouples, and the response is just that patient never got any morphine before. So that is the amazing thing which has, and that we have published almost like 15-20 papers now proving that mechanism. Pardon? Brain research, there are peptides, brain research, there are about 15-16 papers which have been published, maybe 20. So any other question on? No question for Gaylord.
Thank you.
Thank you so much. Let's enjoy the dinner outside and socialize and mix up.